Hello, everyone. Thank you for attending today's webinar, The Future of Vaccine Development and Manufacturing, the Benefits of Combining IDT Biologika's MVA technology platform with innovative analytical methods, presented by IDT Biologika. I'm Andrea Anderson, and I'll be moderating this webinar. I'd like to start off by introducing today's speakers. Nicolas Buch, Senior Expert Development, IDT Biologika. Christina Pospisil, Expert Analytical Development, Cell-Based Bioassays, IDT Biologika. And Dr. Marlene Kochta, Expert Development, IDT Biologika. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few notes before we begin. To access additional resources for today's presentation, please click the Handouts tab button on the left side of your screen. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded and will be available to watch on demand within 24 hours. We'd love to hear from you. During the presentation, please submit any questions you have using the Questions and Answers tab on the left side of your screen. Time permitting, we will conclude with a Q&A session. All right, let's begin. Nicolas, please go ahead.
Today, with my two colleagues, Christina and Marlene, we will present you a quick overview over the benefits of combining our MVA technology platform with its unique innovative analytical methods. But first, I would like to shortly introduce you to IDT Biologika and our general platform approach. IDT Biologika is a leading biologics CDMO providing development and manufacturing of viral vaccines, viral vectors for cell and gene therapy, oncolytic viruses, virus-like particles, and aseptic fill and finish for other biologics with end-to-end capabilities serving companies worldwide. With approximately 2,000 employees and a turnover of nearly 300 million euro, we are working over 3 sites as IDT Biologika under the Glocke Holding GmbH. Our headquarters and GMP facility in Germany is in Dessau, 1 hour south of our process development facility in Magdeburg, which are both less than 2 hours southwest from Berlin. In addition to that, we have 1 US CDC EHS ISO certified site in the US in Rockville, Maryland. We are a full service provider. We cover the entire value chain from contract development through clinical phases 1 to 3 to commercial production of vaccines, cell and gene therapeutics, including quality control, packaging and storage with well-established and sustainable supply chains, including EMEA, FDA, and ANVISA approvals. As many companies, we are developing or having already developed several platforms for different purposes. I don't think I need to dive into the opportunities and challenges behind having platforms. It is clear that having near to plug and play processes in place from development to CTM production allows for fast track turnover time, making it attractive for every company. Our mission is to guide a client from the start of process and analytical development through all clinical phases until commercialization, as well as supporting the industry with capabilities, taking projects in advanced phases like phase 3 or commercialization, having also only drug product and fill and finish projects. In this presentation, I want to touch base on our general platform principles with our recombinant MVA platform as an example for our fit-for-purpose aseptic processes and capabilities, covering strategies to boost viral vectors or viruses like Vaccinia, orthopox virus, CMV, and many others. Where are we right now with our platforms? As you can see on the slide, next to our clients' processes and platforms, we have several platforms built with licensed or even self-owned cell lines. This allows us to support products out of the vaccine and cell and gene business to just catch up on our AAV and lentivirus capabilities. Here I'm talking about generalized process platforms, which can be performed as a bioburden or aseptic process dependence of the construct size and virus type. The reason to choose MVA for this webinar is not only our high expertise, but also based on its size and envelope being very sensitive, resulting in one of the hardest viruses to process under aseptic conditions. We offer a phase 3 ready recombinant MVA cell-stack platform together with an exceptional high understanding of MVA processing. Continuously, we strive to further develop this platform, challenge our own capabilities and technologies. When we speak about MVA manufacturing, we already had to overcome different challenges, starting from specific pathogen-free eggs with our chicken embryo fibroblast technology, transferring the process into our stable immortalized DF-1 cell line on cell stacks, and in the future also in our fixed bed systems. With an average size of around 400 nanometers, the virus does not fit through a sterile filter, making it necessary to run the process fully aseptic. IDT Biologika has a high experience in aseptic processing, making it advantageous for every client with big-size constructs like Vaccinia, CMV, and MVA to use our implemented and validated processing platforms. As a contract development manufacturing organization, we at IDT strive to provide robust and reliable processes for our clients. Therefore, the use of state-of-the-art manufacturing is key. This doesn't only include our upstream technologies. The current downstream processes are based on the tangential flow filtration, while we are currently evaluating the potential of an aseptic chromatography solution for this particular platform. Our GMP bank DF-1 cell line is one option to produce recombinant MVAs with a known end of passaging point approved for CTM production and commercialization, including GMP manufacturing and process performance qualification. This cell line is fully characterized, inclusive its specific amino acid consumption profile, while all necessary analytical assays for a product release are already in place. MVA, an attenuated Vaccinia vector with several vaccine indications, for example malaria, HIV, HBV, and SARS-CoV-2, is also used in several studies as a booster or second shot after, for example, an adenovirus vector vaccination. Besides the clinical data, there are already products on the market like smallpox vaccine from BD and the Ebola vaccine from Janssen. An advantage of the MVA is that it can stimulate both the humoral and the cell-mediated immune response, and that it was indicated in several prime boost regimes being highly immunogenic. It is also well tolerated by immunocompromised people. IDT Biologika itself started to work with MVA in 1997, and we have built up a broad knowledge in the manufacturing of these viral vectors, alone and with partners worldwide. At this point, I would like to show you some data from our phase 3 ready MVA platform on cell stacks. What you see in the diagrams are infectious titers measured by flow cytometry. Christina will explain later, with different MVA constructs and at different scales to show you that the performance of the platform is transferable to different MVA candidates, as well as that it's scalable between different cell stack size. This data shows us that our platform development can take place independent of the recombinant MVA backbone, as we have already tested more than 5 different recombinant MVAs with this platform a small scale of at least cell stack 10 size. In addition, we confirmed the comparability of the process in different scales and with different constructs within our qualified and validated analytical panel to determine impurity profiles linked to residual albumin, DNA, and especially our specific host cell protein assay Marlin will speak about later in detail. But the cell stack platform has its limitation. Although we are able to handle up to 32 CS40s in our GMP suite, equal to approximately 80 square meters of surface, it is limiting us to produce a sufficient amount of dose to cover a vaccine-oriented population. In 2021, we ran a proof of concept study at a third party to become familiar with the scale-up of the universals fixed bed, while we have already a high expertise for scale-up out of pulse iCELLis fixed beds. We want to ensure quick technology transfers to produce batches right first time. Doing the 2 proof of concept runs, the target titer of 1 times 10 to the power of 8 infectious units per milliliter equal to 5 times 6 infectious units per square centimeter were reached, which is approximately 50% of the yield per surface in comparison to the CellStack platform. In 2022, we intensified our process understanding, learned a lot of lessons, and finally tweaked the titer further. Compared the yields over several MVA constructs and run proof of concept downstream runs to establish a scalable and competitive platform able to produce theoretical yields to cover batch size of 50,000 to 100,000 doses. In addition, we are currently implementing Universal's Carbo system in our CTM unit for the first CTM productions. It is important to understand your process and cell-virus interactions during scale-up. It is not just pH, temperature, or dissolved oxygen. A process is never just stirred. Virus infections dependencies are often overlooked in addition to the fixed bed type and its influence through the liquid behavior. We have learned our lessons. We started our development on the PUL fixed bed, which has later shown a lag in virus productivity, although seeing nearly identical growth behavior of the DF-1 cell line itself, independent of the used bioreactor or platform. Nevertheless, this is not the case for every virus reprocessed. Therefore, it is worth it to challenge your system instead of just your critical control parameter. In the diagram, the 3 platforms, CellStack, fixed bed from PUL, and Universals are shown with titers and plug forming units per milliliter on the left and the calculated plug forming units per surface area on the right, showing the different performances of the systems with 3 different MVAs. We are currently in our last phase, nearing our fixed bed yields per square centimeter to the CellStack platform and adapting the new matrix with its very high particle load to the downstream process. Furthermore, we are partnering with different suppliers, testing their prototype, TFF and chromatography columns, narrowing down an idle process for not sterile filterable envelope viruses, which need to process fully aseptically. We did not establish this platform only for Vaccinia and MVA, but have also expanded the knowledge gained to various other projects and virus types our customers can benefit from. For example, understanding the impact of the medium flow through the fixed bed. Is a flow in general impacting infectivity, or is it necessary to stop flows for idle conditions? Last but not least, with a cooperational study on MVA, we tested the so-called virus sensitizer of Virica Biotech within our platform, including the screening of 8 different VSEs, making us familiar with the supplement type. As a result, the combination of 2 VSEs have shown a titer increase of more than 50% in our DF-1 cell line without further process changes. Currently, we are discussing the transfer of the VSEs into our fixed bed process, potentially closing or even exceeding the yield gaps, which we see between the CellStack and the fixed bed platform. It is important for us to think outside the box so far, offer our customers expertise that they may not have shown or known themselves. Therefore, we will test this technology within our cell and gene therapy platforms, especially for AAV on our own HEC-293 cell line as well. Besides our process development expertise, we have implemented fast-track analytical methods to accelerate development timelines on every level. One of them is our FAX method for MVA titer determination Christina will speak about now.
In the scope of innovative analytics, the flow cytometry-based virus quantification enables fast and precise analysis of virus titer samples. But before I explain virus quantification with flow cytometry in detail, I want to give a quick overview on the basics of flow cytometry. A flow cytometer consists of fluidics that direct cell or virus suspensions to a focused light source. The scatter light resulting from a laser striking the cells is transmitted to an electric network by detection optics. The detected and converted signal is hereby proportional to the light intensity. Test material generated with our DF-1 platform process was used to develop an MVA quantification method using flow cytometry. The flow cytometry-based quantification follows the same principles as the conventional methods, as for example the plug assay. Pre-incubated permissive cells are inoculated with different dilutions of a virus suspension. However, different from classical assays, the flow cytometry-based approach relies on the analysis on the single-cell level already during primary infection. For that, cells are stained with a specific fluorescence-labeled antibody. Based on the different fluorescence intensities, populations of infected and non-infected cells can be discriminated. The percentage of infected cells is used for the calculation of the virus titer. A major advantage is that titer results are generated within 24 hours, whereas the conventional plug assay takes 5 days. An additional advantage of this single-cell analysis is the extremely precise quantification of infectious titers. The conventional titration methods, in contrast, rely on manual plaque counting, which is extremely operator-dependent and thus subjective. As you can see in the picture, plaques cannot always be clearly distinguished. Furthermore, it should be considered that not every infectious particle is a plaque-forming one. Both aspects can lead to the underestimation of the virus titer. These differences in assay evaluation are the main reasons for the higher performance of the flow cytometry-based quantification method. Even though the conventional and the flow cytometry assays show a similar repeatability, the intermediate precision results indicate that the flow cytometry-based method is twice as precise. Furthermore, it shows a better recovery, a higher linearity, and offers an operator-independent evaluation. A general comparison of conventional and flow cytometry-based quantification reveals that all are able to measure infectious and absolute titers. Even though the flow cytometry assay requires specific equipment and reagents, it only takes 2 days to acquire titer results, whereas the conventional assays have durations of 5 to 7 days. It furthermore has no limitations in the scope of quantification of lytic or non-lytic viruses or the cell line that is chosen. Quantification in adherent as well as suspension cell lines is possible. For all those reasons, flow cytometry-based virus quantification should be considered as the method of choice for fast release testing and reliable and precise process characterization.
In the previous part, my colleague Christina gave you an insight into rapid titer determination. And now I want to give you an insight into one important safety assay, the HCP ELISA, because HCP removal is one of the major concerns during vaccine manufacturing. HCPs are host cell proteins, which are produced by the host cell system that is used for the production of a biopharmaceutical. Depending on the cell line, HCPs can be a mixture of up to 1,500 different proteins. Several of these proteins are highly immunogenic. They can lead to adverse effects or reduce the efficiency of a product. Wherefore, they pose a high risk for product safety, efficacy, and quality. That is why an important part of the vaccine production is to remove them to appropriate levels and to control them using an analytical method, which is, of course, a very challenging task for an analytical method, as the HCPs vary in different features such as hydrophobicity, isoelectric point, molecular weight, or post-translational modifications, and their expression levels are influenced by the production cell line that is used, the platform vector that is used, as well as the upstream and downstream manufacturing conditions, such as the buffer systems or the equipment. Therefore, a method is needed which is able to detect up to 1,500 different proteins with high sensitivity and specificity, and that is not impacted by the process matrix. The most common method of choice here is the ELISA, which uses a mixture of different antibodies directed against as many different HCP species of the production cell line as possible. If you search the market for anti-DF-1 HCP antibodies, you will not find a vendor, because only antibodies for classical cell lines such as Vero cells or HEK cells are available. This is exactly why IDT invested into the development of specific reagents for a platform DF-1 HCP ELISA, thereby ensuring HCP analysis for IDT's MVA platform. This development was performed in cooperation with Biogenes from Berlin, and in the current and the following slide, the work steps performed by Biogenes are highlighted in yellow, and the ones of IDT are highlighted in green. So the first step was the preparation of an antigen, whereby IDT focused on the fact that it contains as many different DF-1 HCP species as possible. This antigen was then used for the immunization of 28 rabbits, from which the serum was harvested and pooled. Then the anti-DF-1 HCP antibodies were purified from the serum pool, and they were used for the preparation of the capture and detection antibody, which were both characterized. Furthermore, a standard was prepared from the antigen that was used for the animal immunization, and a control was prepared from an MVA-based vaccine. Both materials were also characterized. Then, after approximately 16 months, all the developed reagents were transferred into an ELISA kit production process, as the goal was to have a ready-to-use ELISA kit. As the last step of the reagent development, the ELISA kit was then tested in a real-time stability study over 2 years. After all of the reagents were available, a test procedure was developed at Biogenes, and this assay was initially pre-validated in order to check if the developed assay meets the requirements of the ICH guideline. Then the assay was transferred to IDT, implemented and pre-validated using one of IDT's MVA-based vaccines. So the duration from the start of the antigen preparation until the finalization of the pre-validation at IDT was approximately 2.5 years. Having this platform assay already in place after such a long development time is a great benefit for our future clients, because for future projects, only a product-specific verification of the pre-validated status of the assay is necessary, or in case of higher clinical phases, a product-specific validation. On my last slide, I just wanted to present you the key features of the developed DF-1 HCP ELISA. So the test principle is the sandwich ELISA, which you can see on the left side of the slide. So sandwich ELISA uses capture antibodies, which are coated onto microtiter plates, and they capture the different HCP species from the sample. The secondary antibody is basically the capture antibody, which is labeled with butene, so it also binds to the different HCP species that are already captured on the plate. In the last step, a conjugate antibody binds to the detection antibodies, as it is streptavidin coupled with HRP. The developed ELISA is able to detect 79% of the different DF-1 HCPs from the antigen, which is a very good coverage, and 100% of coverage will never be reached. The ELISA shows no matrix effects, meaning it is not sensitive to the high salt buffers which were used in the MVA production process, and therefore the identified minimum required dilution is 1 to 2. Furthermore, it shows a broad range from 1.5 nanogram per milliliters to 38.3 nanogram per milliliters, and in combination with the identified minimum required dilution of 1 to 2, a very good assay LOQ of smaller than or equal to 3.0 nanogram per milliliters. During pre-validation, the assay showed acceptable precision, linearity, accuracy, and specificity, and can meet the requirements from the guideline. During routine analysis, 5 samples can be tested on one plate within a duration from 2 to 4 days, which is quite a long assay duration, but due to its flexible incubation times, it can be easily performed in parallel to other assays or to other DF-1 HCP ELISA plates, thereby also increasing the throughput. And with this, we're at the end of our presentation. We thank you for your attention and are happy to answer your questions.
Thank you so much for the presentation. We will move on to the Q&A now. As a reminder, please submit your questions using the questions and answers tab at the left of your screen. We have a lot of great questions already, and we will try to get to as many as possible. For the first question, how many different MVA vectors did you test on your phase 3 Ready Cell Stack platform?
For now, we tested 7 different ones on our process development platform, and 2 reached the GMP status, or at least the GMP process.
All right, another question. What is the fixed bed size compensating the lower yields per square centimeter in comparison to the cell stack platform?
So in the beginning, we achieved MVA titers in the fixed bed that 16 CS40s were roundabout equal to 1 Icellus 500. But last year, with further modifications, around 32 CS40s, which would equal 80 square meters, are equal to 1 200 square centimeter fixed bed from Universal. And as I said in the presentation, we are currently tweaking that higher, that the surface area will be equal to each other.
All right, another question. How many samples can be measured within 1 week?
Yes, so for flow cytometry, you can measure approximately 240 samples within 1 week with this assay.
Another question. Does flow cytometry offer further possibilities for the analysis of viral vectors?
Yeah, so you cannot only use flow cytometry for the determination of the infectious titer, but you can also use it for quantification of the insert expression. And since there are flow cytometers that can detect particles up to 80 nanometers, you can also use it for direct particle quantification.
How much sample volume is necessary for running DF-1 HCP ELISA?
Yeah, so using the developed ELISA, we need about 400 microliters of sample volume, as we prepare a 1 to 2 serial dilution, and in total always 4 dilutions per sample, which is why per ELISA plate you can only run 5, but then you're sure that you get a valid result.
Do you have experience using Virica Biotech's viral sensitizers in other platforms?
So far, we only tested this within our DF-1 platform, but we are planning to process this also on our on other platforms, like on our HEC293 cell line for AAV.
Another question, is the transfer of the DF-1 HCP ELISA to GyroLab technology for high throughput analytics possible?
Yes, that is possible. So the capture antibody that has no label, it could be labeled with the Alexa Fluor label that the Gyros technology uses, and the detection antibody, which is now labeled with the biotin, will then be the capture, as the columns in the system, they use streptavidin. So it's possible.
A lot of questions coming in. Here's another one. Do you prefer the Univercells platform over the iCellus?
I mean, in general, they are very similar to each other. We've seen, especially in regards of cell growth, similar results, while the bigger differences are only seen for virus productivity, where one fixed bed can be better than the other. So it really depends on the platform and the target virus you want to process. And therefore, I would always recommend in the beginning to test both platforms.
Another question, would you recommend including the VSE technology for clinical batches?
Mm-hmm. I would say I would recommend it at least to test it. I'm not familiar with the regulatory guidelines behind to test them, but when you are in your first CTM batch, your first clinical material, and you have the chance to use them, and you saw before in your process development that they work quite well, it's probably worth a shot.
What type of challenges have you come across during developing biological vaccines?
I think that's a question for me. And I would say it really depends on the vaccine itself, what kind of virus type we are looking for. If it's a big one, which we have to process aseptic, or if it's small enough to be processed in a bioburden process. So it's a very general questions. There are always a lot of challenges from the yields you want to get out of the upstream, as well as the recovery rates later in the downstream. Hard to answer that questions because there are so many challenges in between. Therefore, that's why it's very nice to have platforms in place where you can also already mitigate your risk.
Have you considered using mass spectrometry for HCP determination?
Yes, of course, we have considered it, and we also do have the technology at IDT, but currently we are staying with the gold standard, which is the DF-1 HCP ELISA. And I mean, development time was about 3 years, so we would also like to keep using it, and we would also like to to perform process characteristics or characterization with mass spectrometry, but as additional information, basically.
What type of chromatography do you plan to select in your MVA platform process? Also, which type of matrix will you use?
Very good questions. We're in the middle of planning these experiments or have already done some, but based on confidential reasons, I can't answer these questions properly.
As the HCP coverage is 79%, not all proteins will be captured. Does the 79% refer to the different DF1 proteins or to the amount of protein detected?
So that's correct. Coverage is 79%, and that means so only 79% of the antigen that was used for the animal immunization, which was basically a pool of a lot of different DF1 HCP proteins. And now the antibody mixture that was produced by the animals, it is only able to capture 79 of the 100%. So you never have the sufficiency that the animals will express antibodies in the same levels against all of the different proteins. Like I mentioned, it's up to 1,500 different proteins. They have different expression levels, and it's very hard to capture all of them. That's why it was very important that that we took our time during the antigen preparation. So it was about half a year, and we have performed, I think, like 15 preparation rounds in order to have a high variety of all the different kind of HCPs in the antigen mixture. And then we used 28 rabbits. So every rabbit also has another immune response against certain other HCPs. So you try to get as much variability as you can, and still you only get 79% of the coverage, which is very good though. It's very high. So
How do the titers differ between the two assays, traditional plaque versus flow cytometry based?
Yeah, so from experience, it's about half to 1 log higher in the flow cytometry-based assay, but it really depends on the traditional plaque assay you use, because between the plaque assays or TCID50 assays themselves, there are also differences. So to have an exact value, you need to do a comparability study between the assays, but it's approximately half to 1 log.
Regarding the virus titer assay using flow cytometry, how do you label the virus?
Yeah, we stain with a directly fluorescence labeled antibody against the virus.
Another question, can the services for titration or HCP ELISA be ordered as separate services, like without GMP production?
So Marlene, I would answer that. So currently we only
yes.
do in-house analytics. So no, at the moment it's not available.
We have one more question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. So for this final question, have you compared virus production between cell factories, fixed bed reactors, and microcarriers?
So for our DF-1 cell line, we have only comparability data between cell factories and cell stacks and fixed bed reactors, due to that they didn't like to grow on the microcarriers. They were too sensitive, that we stopped early process development in the last years. For that, but we have data for other platforms, but often these are then client-related platforms, and comparability studies I'm not allowed to talk about.
Well, thank you all for attending this Fierce Biotech webinar and for submitting so many great questions. I'd like to thank our speakers for participating and IDT Biologika for presenting today's webinar. A recorded version of this webinar will be available for you to access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.